So this video is a brief introduction to metabolism and regulation of metabolism. We will be focusing mainly on regulation of metabolism. If you feel that I am speaking slowly, you can increase the playback speed at your end. Reactions inside cells rarely occur in isolation. They occur in organized multi-step sequences called pathways, where product of one reaction is substrate for another reaction. Pathways can be catabolic, degradative, or anabolic, synthetic. Different pathways, they intersect, forming integrated purposeful network of chemical reactions. Metabolism is the sum of all chemical changes occurring inside a cell. Metabolome is the complete collection of all small molecules called metabolites found within a cell, tissue, organ, or organism. Coming to catabolism, it occurs in three stages. Proteins, carbohydrates, fats are broken down into their monomers, amino acids, monosaccharides, and glycerol and fatty acids. They are eventually broken down into acetyl-CoA, which goes to TCA cycle to form ATP. And this ATP, this is catabolism. And when ATP is used for synthesizing proteins, polysaccharides, lipids, nucleic acids, that is anabolism. Anabolic pathways generally utilize NADPH as the donor of reducing equivalents. Metabolic pathways must be coordinated so that the production of energy or synthesis of end products meets the demands of the cell. This regulation can be via intracellular communication or intercellular communication. Intracellular communication is the rate of the reaction being influenced by availability of substrate. That is, if the substrate is in less quantity, it can inhibit the reaction. If the substrate is in excess, it can make the reaction happen faster. If the product accumulates, it can inhibit the reaction. That is called as product inhibition. Reaction rate can be influenced by alterations in the level of allosteric activators or inhibitors. Intracellular signals elicit rapid response. They are important for moment-to-moment regulation of metabolism. Intercellular communication is slightly slower than intracellular signals. However, it is essential for development and survival of organisms. We are multicellular organisms. We need intercellular communication. And this can be achieved by direct surface-to-surface contact, or it can be achieved by something called as gap junctions, where the cytoplasm of the adjacent cells is in direct communication with each other, or it can be by chemical signaling through hormones or neurotransmitters or autocoids, paracrine signaling or autocrine signaling. And these hormones or neurotransmitters, they act via second messenger systems or receptor tyrosine kinase, that is enzyme linked to the receptor, or they act via ligand gated ion channels, or they act via nuclear receptor. In this video, we'll be dealing with second messenger systems and receptor tyrosine kinase. So these are the four general types of signal transducers. You can pause and read the diagram. Out of this, we'll be discussing the first two. What are second messengers? They are basically like a relay. They are molecules that intervene or act as mediator between original extracellular signal, hormone or neurotransmitter, and intracellular effect. They are part of a cascade of events that transduces ligand binding to intracellular response. Two most widely used or most widely recognized second messenger systems are adenylyl cyclase or adenylate cyclase system and calcium phosphatidylinositol system. Both of these involve ligands binding to specific G protein coupled receptors or GPCR on the plasma membrane. G protein coupled receptor consists of extracellular ligand binding domain and they have 7 transmembrane alpha helices 1, 2, 3, 4, 5, 6, 7, an intracellular domain which acts with trimeric G proteins. This intracellular domain. Adenylyl cyclase system or cyclic AMP second messenger system is a membrane-bound enzyme. Adenylyl cyclase is a membrane-bound enzyme which converts ATP to 3' 5' adenosine monophosphate or cyclic AMP. Chemical signal, that is the hormone or neurotransmitter, it can increase or decrease adenylyl cyclase activity. Each chemical signal can bind to binds. Each chemical signal binds to a unique G protein coupled receptor. So these are the steps in cyclic AMP second messenger system, and these are for a stimulatory signal. That signal will activate adenylyl kinase by G alpha protein that will cause cyclization of 5' AMP to cyclic AMP. That will activate cyclic AMP dependent protein kinase A, which causes phosphorylation of substrate protein, bringing about intracellular effect of the signal. And when it's time for the signal to stop, there is degradation of cyclic AMP by phosphodiesterase, and this inactivation of cyclic AMP dependent, and then that causes inactivation of cyclic AMP dependent phosphoprotein kinase A. Dephosphorylation of substrate protein by constitutively active phosphatases, and termination of intracellular signal. We'll understand in the next diagram as well. So, effect of activated occupied G protein coupled receptor on second messenger formation. Second messenger here is cyclic AMP. This is mediated by G proteins. So G proteins have 3 subunits, that is, it is trimeric, alpha, beta, gamma, and they are called G proteins because the alpha subunit binds to GDP or GTP to form a link between receptor and adenylyl kinase. So in the inactive form, alpha subunit is bound to GDP. You can see here it is bound to GDP, alpha subunit. And when the ligand binds, that is the hormone or neurotransmitter binds to the external surface, that causes a change, conformational change in the receptor, causing replacement of GDP with GTP. So now on the alpha subunit, GDP has been replaced by GTP. And the GTP bound form of alpha subunit dissociates from beta and gamma subunits, and it moves to adenylyl kinase, affecting the activity of adenylyl kinase. So this alpha subunit, you see, it has broken away from the beta gamma and it has gone to adenylyl kinase. It can affect the activity of adenylyl kinase. That means it can either increase its activity or decrease its activity, depending on whether that alpha subunit, G alpha protein, is stimulatory or inhibitory. If it is stimulatory, it is called as GS. Inhibitory, it is called as GI. And so let us say that it activated adenylyl kinase, causing cyclic AMP, and then uh, there is this phosphodiesterase. Oh, we'll come back to that later. So, in absence of signal, GTP gets hydrolyzed to GDP due to inherent GTPase activity of G alpha. G alpha dissociates from adenylate cyclase and reattaches to beta gamma subunit. So, this in the inactive form, GDP is bound to the alpha subunit. When the ligand, hormone or neurotransmitter binds to the external surface, GDP gets replaced by GTP. Then this alpha subunit detaches from beta gamma and goes to adenylate cyclase and either activates or inhibits it. And this alpha subunit has inherent GTPase activity. So after some time, this GTP by itself it cleaves to GDP. And when it gets cleaved to GDP, this alpha subunit goes and binds to beta gamma subunit. And that brings the action of the hormone or neurotransmitter, that terminates the action of hormone or neurotransmitter. And obviously, when this alpha subunit will detach from adenylate cyclase will also, the inhibition or increase in activity on adenylate cyclase will get reversed, and so whatever action it had on adenylate cyclase further downstream, those actions also will get reversed. So whether it increases or decreases adenylate cyclase activity, once let us say that it increases adenylate cyclase activity, so more of cyclic AMP will be formed, and when it is formed, this cyclic AMP activates protein kinase A by binding to its regulatory subunit, causing dissociation of catalytic subunits. So protein kinase A is a tetramer. There are two catalytic subunits and two regulatory subunits. When catalytic subunits are bound to regulatory subunits, protein kinase A is inactive. Protein kinase A is dependent on cyclic AMP to become active. What does it mean? When this cyclic AMP binds to this protein kinase A, the catalytic subunits will dissociate from the regulatory subunits, and then these catalytic subunits can phosphorylate the substrate protein, right? And the phosphorylated protein then brings about the intracellular effects of that hormone or neurotransmitter. Catalytic subunits transfer phosphate from ATP to specific serine threonine residues of substrate protein. Phosphorylated substrate protein can act directly on cells' ion channels, or if the phosphorylated substrate protein is an enzyme, it can get activated or deactivated. Cyclic AMP phosphodiesterases cleave cyclic AMP to 5' AMP to terminate the effect of the signal. Degradation of cyclic AMP leads to reassociation of catalytic subunits of protein kinase A with regulatory subunits and inhibition of protein kinase A. Right? In the event of decreased activity of protein kinase A, protein phosphatases, which are constitutively active, they dephosphorylate substrate protein, thus terminating the effect of extracellular signal. So there are phosphodiesterases which the cell uses to control this effect of the signal. Phosphodiesterases will cleave cyclic AMP to 5' AMP, and if cyclic AMP is not there, these catalytic subunits of protein kinase A will bind to regulatory subunits and protein kinase A will become inactive. That means the protein which it was phosphorylating, it won't be, the phosphorylated protein then will be cleaved by protein phosphatase, which is constitutively active. Matlab it is active all the time. But because of this protein kinase A not being there to phosphorylate it anymore, whatever was phosphorylated by protein kinase A, that also gets dephosphorylated by protein phosphatase, and then that effect of extracellular signal gets terminated. This is another diagram. Say in here it is showing example of epinephrine binding to its specific receptor, right? Beta adrenergic receptor. Allosteric change in hormone receptor complex causes GDP bound to GS alpha to be replaced by GTP, activating GS alpha. Activated GS alpha separates from GS beta gamma, moves to adenylyl cyclase, activates it. Many GS alpha subunits may be activated by one occupied receptor. This is important to understand. Adenylyl cyclase catalyzes formation of cyclic AMP. Cyclic AMP activates protein kinase A. Phosphorylation of cellular proteins by protein kinase A causes cellular response to epinephrine. Cyclic AMP is degraded by cyclic nucleotide phosphodiesterase, and that reverses the activation of protein kinase A and terminates the signal. This is one example. We'll see there is one beautiful article on PubMed on second messengers. I'll share that article link in the description. The signal is always amplified by second messenger. So epinephrine triggers a series of reactions in hepatocytes in which catalysts activate catalysts, resulting in great amplification of original hormone signal. The numbers of molecules shown are illustrating the amplification, and they are just an estimate, right? Binding of one molecule of epinephrine to beta-adrenergic receptor on the cell surface activates many, possibly hundreds of G proteins, one after another, each of which go on to activate molecule of an enzyme adenylyl cyclase. Adenylyl cyclase acts catalytically, producing many molecules of cyclic AMP for each activated adenylyl cyclase. Because two molecules of cyclic AMP are required to activate one protein kinase A, this step does not amplify the signal. So one epinephrine molecule is stimulating several GS molecules. They are activating a lot more cyclic AMP. They're leading to a lot more, like almost 20 times more cyclic AMP formation. And those cyclic AMP in turn are activating more of protein kinase A molecules. And then they are phosphorylating protein kinase A, lot of phosphorylases, the enzyme or the substrate protein, and then that substrate protein is having effect, like here this protein kinase A phosphorylase, right? Glycogen phosphorylase is active in phosphorylated state. So phosphorylation of glycogen phosphorylase activates glycogen phosphorylase, and that leads to, right, uh, glycogenolysis and formation of glucose. So just one epinephrine molecule led to approximately 1 lakh glucose molecules being released into the bloodstream. That is the amplification achieved by second messenger systems. So, clinical implications: cells use phosphodiesterases to determine how long a signal lasts. Phosphodiesterase inhibitors are used to prolong the effect of cyclic AMP. And there are a wide variety of clinical implications, including treatment of erectile dysfunction, asthma, COPD, managing heart failure, peripheral artery disease, autoimmune disorders like psoriasis, psoriatic arthritis, or immune mediated disorders like atopic dermatitis. Cholera toxin increases cyclic AMP by covalent modification, which inhibit the GTPase activity of G alpha, and so the GTPase activity is not inhibited. So G alpha will keep on stimulating adenylyl cyclase. Cyclic AMP will keep on increasing. This causes secretory diarrhea. In Bordetella pertussis or whooping cough, cyclic AMP increases because of inactivation of inhibitory G alpha. So there is no one to inhibit that GTP, the adenylyl cyclase. So there is increase in cyclic AMP, which causes paralysis of the cilia, leading to accumulation of respiratory secretions and cough. Coming to calcium phosphatidylinositol second messenger system, also called as IP3DAG system or inositol triphosphate diacylglycerol cell signaling. So it is also a G protein coupled receptor, a G protein coupled receptor mediated. So this hormone binds to G protein coupled receptor that activates the receptor, and you know that the alpha subunit, then GDP gets replaced by GTP and it dissociates, and in this case it binds to phospholipase C, the alpha subunit. This phospholipase C cleaves inositol 4,5 biphosphate, inositol 4,5 bisphosphate to inositol triphosphate and diacylglycerol. Inositol triphosphate binds to receptor on smooth endoplasmic reticulum and causes release of calcium from smooth endoplasmic reticulum, and this calcium and diacylglycerol together activate protein kinase C, which phosphorylates the proteins which are required for intracellular effects. So protein kinase A was cyclic AMP dependent. Protein kinase C is dependent on calcium and diacylglycerol. So, and when the signal needs to be terminated, this phospholipase, this inherent GTPase activity will convert this GTP to GDP, and this will go back and the signal will be terminated. Clinical implications of this are: these IP3DAG receptors are primary target for antihypertensives like calcium channel blockers, Prolonged activation of this pathway by angiotensin II contributes to pathological myocardial remodeling and heart failure. Overactive IP3DAG is implicated in bipolar disorder. This mood stabilizer lithium dampens the overactive IP3DAG signaling. In obesity, chronic elevation of intracellular calcium dampens IP3DAG signaling, causes insulin resistance. Immunosuppressant cyclosporin used to prevent graft rejection relies on dampening IP3DAG of T lymphocytes. This is another diagram showing the same thing. You can pause and check it out. So there is this article, that link I'm sharing in the description. I just want to read some things from this article. Second messengers are small molecules and ions that relay signals which are received by cell surface receptors to effector proteins. Second messengers are typically present at low concentrations in resting cells, very important to note, and they can be rapidly produced or released when cells are stimulated. Levels of second messenger are exquisitely controlled, such that these messengers then diffuse rapidly from the source and bind target proteins and alter their properties to propagate signaling. Cell surface receptors signal effector proteins via second messengers. Second messengers fall into four classes: cyclic nucleotides, cyclic AMP, lipids, diacylglycerol, ions, gases, and free radicals. We just saw adenylyl cyclase and IP3DAG, but there are these other two types also: ions, gases, and free radicals. Signals received by receptors at the cell, or in some cases within cell, are often relayed throughout cell via generation of small rapidly diffusing molecules, which are referred to as second messengers. These second messengers broadcast initial signal, the first message that occurs when a ligand binds to a specific cellular receptor. Ligand binding alters the protein conformation of the receptor such that it stimulates nearby effector proteins that catalyze production, or in case of ions, release or influx of the second messenger. The second messenger then diffuses rapidly to protein targets elsewhere within the cell, altering activities as a response to the new information received by receptor. Second messengers disseminate information received by cellular receptors rapidly, faithfully, efficaciously. They are small non-protein organic molecules or ions that bind to specific target proteins, altering their activity in a variety of ways, allowing them to respond appropriately to information received by receptors. Key advantage, this is very important. A key advantage of second messengers over proteins. Why does cell not use proteins as second messenger? Is that second messenger levels are controlled with rapid kinetics, whereas it may take tens of minutes for levels of protein to increase significantly. Most second messenger levels increase within microseconds, like ions, to seconds, like diacylglycerol. They are often produced from precursors that are abundant in cells released from stores that contain high concentration of second messengers, or so their generation is not rate limiting. Thus, when appropriate signal is received, second messengers are rapidly generated, diffuse rapidly, and alter target protein function highly efficiently. Tyrosine kinase receptors are used by insulin or growth factors. Receptor tyrosine kinases are cell surface transmembrane receptors that regulate vital processes like cell growth, survival, division. Each receptor is made of two extracellular alpha subunits which bind to the ligand, and two transmembrane beta subunits which have tyrosine kinase activity. We'll see that in the diagram later, but these are the steps. These are the steps by which uh, the second messenger, it's just not a second messenger system. These are the steps by which tyrosine kinase receptor signal is transferred. So the ligand binds to the receptor outer domain. This causes dimerization of the inner domain, right? And that dimerization leads to autophosphorylation. It pulls the intercellular kinase domains together. Receptor undergoes autophosphorylation where phosphate groups are added to specific amino acid residues. We'll see this in the later diagram. Docking of effector proteins. The phosphorylated tyrosine kinase residues acts as high affinity docking sites. Intercellular relay proteins with specific domain bind to these sites. Once bound to docking site, adapter proteins become active and they trigger vast downstream networks like MAP-ERK. This is also called as mitogen activated protein kinase extracellular signal regulated kinase, mitogen activated protein kinase extracellular signal regulated kinase pathway, or phosphoinositide 3 kinase and protein kinase B is AKT pathway, or JAK-STAT pathway, which is also called as Janus kinase, and signal transducer activation of transcription, signal transducer and activator of transcription pathway. So basically this, then there are several downstream pathways that can be activated, which direct the cell proliferation, metabolism, and survival. You can see here that binding of the ligand causes these beta subunits, the intracellular domains, to come together and causes autophosphorylation, and that leads to all of these signal transducers will cause these pathways to get activated, and that will lead to all of these effects like cell proliferation, cell growth, differentiation, survival, adhesion, migration. This is a good diagram. So these are the alpha subunits which are extracellular, and these are the beta subunits which have a transmembrane component and an intracellular component. So insulin binding region of the insulin receptor lies outside the cell, comprises two intertwined alpha subunits. Binding of a single molecule of insulin is communicated through the single transmembrane helix of each beta subunit, this, to the paired tyrosine kinase domains inside the cell, moving them towards each other and activating them to phosphorylate each other on 3 tyrosine residues. These uh, tyrosine kinase domains, they were apart, but binding of insulin causes them to come together and activates them to phosphorylate each other on 3 tyrosine residues. And then the introduction of 3 highly charged phosphorylated tyrosine residues forces a change in the position of the activation loop away from the substrate binding site, which thus becomes available to bind and phosphorylate a target protein. So this activation site, triply phosphorylated activation loop moves dramatically, making room for target protein in substrate binding site. This is the docking site that we were talking about. And then the relay proteins, they bind to these docking sites and they become active after binding to the docking site, and they trigger downstream networks like these MAPK, ERK, PI3, AKT, JAK-STAT, and they lead to the outcome like cell proliferation, growth, differentiation, survival, adhesion, migration. Clinical implications of this is that because tyrosine kinase receptors are used by growth factors, their overactivation can lead to tumor, and anti-cancer, anti-tumor drugs target these receptors to shrink the tumor. Defects in insulin receptor autophosphorylation leads to insulin resistance and type 2 diabetes mellitus. Fibroblast growth factor receptor FGFR3 mutation leads to achondroplasia. Achondroplasia is the most common cause of dwarfism. So we'll discuss nuclear receptors and ligand-gated ion channels in some other lecture. Thanks, that is enough for today. Have a great day ahead.